Okay, so it seems like we can start. So welcome everyone to the to our fall semester series of tissue talks. We were looking back and realizing it is now the beginning of the sixth year. So many of you know we started this during COVID, to when COVID hit us, to keep like the sense of purpose and connectiveness. And both Navid and I expected the tissue talks to live for maybe a few months. We were very naive in this expectation. And so the COVID lasted for a long time. But then, I mean, we had like a really good response to this seminar. So we just decided to keep it going. And you will see this semester brings us some fantastic speakers, starting with Professor Roger Kamm today. Just a brief reminder, please you can post your questions during the talk, but please use the Q&A feature, not the chat, to do that. And at the end, Professor Kamm will be answering as many questions as possible. So I think most, if not all of you, know Roger Kamm very well. He's a real leader in our field, but I will still introduce him briefly. Professor Kamm is Cecil and Ida Green Distinguished Professor of Biological and Mechanical Engineering at MIT, Massachusetts Institute of Technology. He trained at Northwestern University by obtaining bachelor's degree in mechanical engineering and then moved a little east to MIT to obtain his master's degree and PhD degree in mechanical engineering. He is, in addition to being distinguished professor, he is also director of the MIT NSF Science and Technology Center for Emergent Behaviors of Integrated Cellular Systems. This long name is very descriptive of the essence of his research. His laboratory, his research team has been focused for a long time on how the fundamental concepts in mechanics, both fluid and solid mechanics, can be applied towards advancing our understanding of biological and physiological phenomena in cells and tissues. His studies range from respiratory organs, lung, to eyes and cardiovascular systems. So it's very broad, and this tells us a lot about the conservation of some of the principles and approaches. I think it's safe to say that Professor Kamm has really made major advances in bridging the fields of mechanics with biology and chemistry. He has worked at the interfaces of disciplines for decades now, and he was very successful in building realistic environments as models for studies of cellular and tissue biology and function. In recent years, he has published some seminal work in vitro, developing in vitro models of metastatic cancer and in vitro models of different neurological diseases, ranging from Alzheimer's to dysfunction, to motor neurons and diseases based on dysfunction of neuromuscular junctions. He is also a fantastic collaborator, which I can testify about firsthand. It's really a pleasure to work together. His work has had a lot of traction. I mean, people are really paying attention and reading his papers. He published over 270 research articles that are peer-reviewed and accumulated over 60,000 citations with a very impressive H index of 133. Not surprisingly, he was broadly recognized for his contributions. He is a fellow of several major societies, honorific and professional societies, such as American Institute for Medical and Biological Engineering, American Society of Mechanical Engineers, and Biomedical Engineering Society. In 2010, he received the ASME Listener Medal, and 5 years later, he received the Hiskis Medal from the European Society of Biomechanics. It's important, these big awards are for lifetime achievements in his field. He was elected a member of the National Academy of Medicine in 2010, and a little bit more recently, he was also elected to the National Academy of Engineering, which is a major accomplishment. His work is also highly translational, and this is reflected in two companies that he has founded. Roger, thank you so much for being with us today. And you can now share screen and start your slides.
Okay, great. Thank you for that kind introduction, Gordana. Let me just find my presentations.
Sure.
There, I hope you're seeing the right screen.
Yes, it looks great.
Okay. So again, thanks for the invitation to give this talk. I spoke at this series several years back on a related topic, but somewhat different. And I look back at my earlier presentation, and I was glad to see that I think there's one image that's in common. So most of you should be seeing this for the first time. So I'm going to talk about the work that we've been doing in developing these in vitro models for first the blood-brain barrier, but then also combining them with cerebral organoids to develop models for Alzheimer's disease. I don't think I need to tell anybody here how important and devastating Alzheimer's and Parkinson's are as a disease for the elderly. Has a huge impact on the quality of life. Tremendous burden, economic burden to the families that are affected. And we still don't really understand the factors that lead to the pathogenesis of the disease, and certainly don't know how to cure it. There's tremendous work going on in developing new therapies, but it's difficult to develop new ones simply because the models that we have limited validity and are not very predictive. That isn't to say there aren't a lot of drugs in the pipeline. This is a map from just a paper about a year ago that shows all the different drugs that are being developed for Alzheimer's. The ones I'll draw your attention to are the disease-modifying biologics and the disease-modifying small molecules here. In these, you can see that there are 164 trials that are assessing 127 drugs in the current drug pipeline. 15 of these target amyloid beta and 7 target the tau tangles. But the models that we have for screening for these different drugs is really very limited. And the human trials, of course, are extremely lengthy, and unfortunately, they often end in failure. And I think probably most of you know, to date, we only have 2 disease-modifying drugs that have been approved and that are on the market. So a little bit of background about Alzheimer's, and in particular neurovascular disease, and how that's thought to be connected to Alzheimer's. That's going to be the main focus of my talk today. Some of the pathological hallmarks of Alzheimer's are brain atrophy. You get accumulation of amyloid beta plaques around the vasculature. Here are some images of that. And also what are called neurofibrillary tangles, tau tangles that accumulate in the brain. In terms of the neurovascular dysfunction, we see that there's impaired barrier integrity. So in other words, the vessels become leakier as a result of the amyloid deposits around the vessels. And also what we find is that the inflammation that's often associated with Alzheimer's, that the clearance of A beta seems to be impaired. So there's a real need to have these integrated experimental models that allow us to look at the interaction between the vascular system and the neurons and astrocytes in the brain, and how they crosstalk and how they influence the progression of the disease. So what are the models that we have for looking at brain and modeling brain tissue? Most of the work that's been done so far has been done in animal studies, either in mouse or in primates. And of course, along with these are various two-dimensional in vitro assays that have been developed over the years. But more recently, and I'd say over the last 10, 15 years now, we've now been able to generate cerebral organoids. Those are now coming into common practice for at least the modeling of disease processes and have potential, I think, for drug screening. And then we also have these microphysiological systems, or what are also sometimes called complex in vitro models. I'll probably use the two terms interchangeably. And that's really what I'm going to be talking mostly about today. And really the combination of organoids in a system like this, where we can model the disease process and potentially use this as a platform for screening for new therapies. The other thing that's been happening in the background is that there has been a big push to get away from animal models, both mouse and primate. Just in this year, in April, the FDA announced that they were going to phase out animal testing requirement for monoclonal antibodies and other drugs. A little bit later, in July of this year, the NIH announced that it would no longer award funding to new grant proposals solely relying on animal testing. That's going to have huge impact, I think, going forward on how we can actually study and develop new therapies, preclinical studies for new drugs. So as a result, these complex in vitro systems and also computational approaches are going to be relied on to an increasing extent for drug development. And of course, aside from the animal welfare concerns, it should also be noted that these non-primate models of Alzheimer's and Parkinson's really are very poor predictors of efficacy in disease. And that's one of the reasons that so many of the drugs that enter into the drug development pipeline in the end fail, and we have so few today. So what are the different approaches that people use, in vitro approaches? We have membrane-based models. These are like transwell systems that you see in the upper left-hand corner, where you might have endothelial cells plated on one side of a membrane, pericytes on the other, and then astrocytes and maybe neurons in a separate chamber. You also have these compartmentalized models where maybe there isn't a membrane separating them so that the cells can interact in a more natural way. The endothelial cells here, maybe the neurons and astrocytes in this region, and then just these regions just separated by pillars that allow you to put a three-dimensional gel in those compartments and to culture them. Also, there are a number of spheroid-based models where we can have, for example, brain organoids that can be placed and they can interact with each other, in some cases coated with endothelial cells in order to make the neurovascular coupling. And then finally, there are the self-assembled models, mainly the type that I'm going to be talking about today, where these are self-assembled models containing multiple cell types that are present in the blood-brain barrier. So some of the advantages of these self-assembled models is that they do reproduce the natural process of vascular development. So the vessels are actually grown by natural self-assembly. You can have direct cell-to-cell contact and interactions between, for example, the endothelial cells, the pericytes, and the astrocytes. Good physiological morphologies. They recapitulate to a large extent what you see in the brain, along with good functionality in terms of permeability and other functional measures. And finally, we can use these for long-term, mid to long-term studies. And some of our studies have gone out to about a month, which is really necessary in order to capture the disease phenotype. So the types of model that I'm going to talk about today, here's the basic platform. They essentially consist of a central gel region, sort of the yellowish region that you see here. The blue media channels flanking on either side so that you can control the delivery of different chemicals, generate pressure gradients, generate flows through the cells that are plated in the yellow region. That's where the blood-brain barrier is going to be in our model. And then oftentimes we have this center port, which is a place where we can insert, for example, brain organoids. So you can get the coupling then between the cerebral organoids or the brain organoids and the BBB. I also want to mention that and disclose that I am a co-founder of a company, AIM Biotech, that manufactures devices like this. We also have industrial funding from the companies that are listed here: Amgen, AbbVie, Novartis, Takeda, and Daiichi Sankyo. So what can you do with platforms like this? First of all, you have good imaging capabilities. So immunofluorescence is a major tool that we use, confocal imaging, reflectance imaging. You can use TEM, do time lapse. Most of the biochemical analyses that you'd like to do that are traditional to use in, say, transwells or multi-well plates, ELISA, Luminex assays, RT-PCR, RNA-seq, and so forth. These can all be done with the cells that are extracted from systems like this. But then we can also do functional assessment or functional assays. And in this particular case, looking at neurological function using, for example, MEAs, measuring permeability, the leakiness of the vessels. These are some data that from a recent review. And then in other studies, we look at cell migration, tumor cell extravasation, and so forth. So there's a lot that you can do still staying in essentially the same platform. I'm going to be talking primarily today about the applications to the brain. We do have a publication that came out just this past year, during the past year, where we developed a microphysiological model that incorporated, in this case, a non-isogenic blood-brain barrier with embryonic stem cell-derived neural tissue. And over the left-hand side, you can see this is basically the gel region in the device that I just showed you. Here's where the neurons are placed, and then we have access on the two sides so that we can generate a pressure gradient and drive flow through the vascular network. This is just a blow up. This is a case where we have Alzheimer's expressing neurons expressing various mutations associated with familial Alzheimer's disease. And you also have the blood-brain barrier around all around these neuron clusters. So you can see you get good interaction between them. Anything that's secreted by the neurons will influence and migrate out into the BBB, and any factors secreted by the blood-brain barrier cells will also enter into the spheroid. In this model, what we did is we used the two types of neurons, one a control and then one that carried these various VAD mutations. And we cultured these over a period of 4 weeks. And on the left-hand side, what we're looking at is just the vascular structures by themselves. And you can see the endothelial cells forming into this three-dimensional perfusible vascular network. But you don't see any of the 3D6 stain, signifying that there's very little amyloid beta accumulating in the control case. Whereas in the situation where we use neurons that overexpress amyloid beta, you can see here that the percentage of A beta coverage in the blood-brain barrier in the model comes up to about 10% in the AD case. We could also show in the same model that there are other changes in phenotype that were associated with the disease process. One is that we found that the pericyte coverage tends to be reduced between the control and the AD. Claudin 5, which is one of the main junctional proteins in the endothelial monolayer, the expression is reduced in the Alzheimer's case relative to the control case. And then finally, associated with that, the permeability to, for example, a 3 kilodalton dextran increases. So all of these are typical phenotypes associated with neurovascular disease. The limitations of this model, though, are one that it's non-isogenic. And one of the problems with that is that when you use primary cells, we found that every time you replaced one cell type, and now we're talking about 4 different cell types in the model, we had to re-optimize the model. So it took a tremendous amount of time. what we're looking for is to have a model that would be consistent year after year, that we could derive entirely from iPS-derived cells. Also, the model lacked some critical cell types. For example, we didn't have any microglia. Microglia are known to interact with the blood-brain barrier, and we felt that it was a shortcoming of the model that we didn't have them at the time. The donor-to-donor variability in primary cells is obviously an issue I just mentioned. And also the model was, at that point, at the time of publication, we had done some characterization, but not very extensive characterization. So in the studies that I'm going to present to you today, we've tried to address these concerns in the model in order to put it up a notch, make it a little bit more rigorous, more predictive, and more consistent. So I'm going to talk in two parts. The first part of my talk is going to be in talking about the isogenic, fully iPS-derived model of the human blood-brain barrier model that we have. And then I'll talk about combining that with human cerebral organoids to develop a model of neurodegenerative disease, and then to look at the phenotypes of that disease in vitro. So starting out with the BBB, this is basically a cartoon of the structure in the small vessels in the brain. You have the endothelial cells that line the lumen. You have pericytes that tend to wrap around that lumen. And then you have astrocyte end feet. These are projections from the astrocytes that tend to interact, communicate with and interact with the endothelial cells and the pericytes. So that structure, we feel, is important to reproduce in any model that's going to be able to reproduce function and recapitulate function of the blood-brain barrier. So for the iPS-based model, we worked with two colleagues here at MIT, Rudolf Janisch and Maya Mitalipova at the Whitehead Institute to develop, in this case, an Alstem 11 iPS cell line that we could use to derive our endothelial cells, and also trying a couple of different astrocyte differentiation protocols in order to get our astrocytes, and then the pericytes that we use are also obtained from a protocol actually developed by the Whitehead. The model that we're using is similar to the one that I just showed here, the media channels on the side. The hydrogel chambers where we seed the multiple cell types for the self-assembly of the blood-brain barrier model. So I'm going to go through the protocols of each of these very briefly. First of all, for the endothelial cells, we use an inducible ETV2 protocol that's shown right here. It's a short protocol. It's only about 5 days. But we have a 1-day DOX induction of ETV2, activation of ETV2, which gives rise to a very high yield of endothelial cells. We don't have to sort these. We can expand them immediately after the 5 days. We get a large number of cells, but not only a high yield, but they can also proliferate very effectively. And then if we do the RNA-seq analysis, when we profile these cells and do principal component analysis, what we find is that the cells that we derive from the iPS-11 cell line clusters reasonably closely with the primary human brain microvascular cells. And I'll show you that in this slide. So this is some of the RNA data that we've obtained. This is just a selection, a subset, but I wanted to highlight a couple of features here. One is that if we look at the different transporter proteins, which are the ones you see over here on the left, a lot of those that are specific to the brain tend to be expressed in about the same level in the human brain microvascular endothelial cells and the iPS-11 ETV2 cells. If we look also at canonical markers for the endothelium, for example, VE cadherin and PCAM1, we see high levels of expression of those two in the cell line. And also we wanted to look at epithelial markers because other cell lines that had been proposed as brain microvascular endothelial cells actually wound up having somewhat of an epithelial phenotype. We find virtually no expression of EpCAM in these cells. So they really are true endothelial cells. We're in the process of profiling these in 3D now. This, by the way, is all based on 2D comparisons, but we should have the 3D data within a month or so. I also wanted to point out that a lot of the key A beta transporters, for example, the BCRP, LRP1, and RAGE, are expressed in these ETV2 cells, as you see over here. So getting back to the astrocytes, first of all, this is the work that was done in collaboration with the Whitehead. We tried two different differentiation protocols. One was published by Perio in 2018. Another is a differentiation kit provided by Thermo Fisher. Although the Thermo Fisher differentiation protocol is somewhat shorter, it's only about 4 weeks. What we found is that the Perio protocol worked better for us, and I'll describe why. Part of the reason we chose to use that is that we wanted to get away from the use of FBS, which is used in the Thermo Fisher protocol. And you can see over here just some illustrations, some immunofluorescence images in 2D of the astrocytes that are developed with the two different protocols. For the pericytes, we had tried two different protocols developed by the Whitehead. One is a mesodermal pericyte protocol. So this is a generic pericyte. And then we compared that to a protocol that differentiates the cells through a neurocrest phenotype. So they're more brain specific. So we call these the neurocrest cell pericytes. And here again, when you do the profiling of these, what you find is that here are the brain primary pericytes, and they cluster fairly closely with the IPS-11 derived neurocrest cell pericytes here. So we're pleased with the protocol there. And again, this is with a defined and serum-free medium, so getting relatively better consistency. This is what they look like when plated out in 2D, and they express most of the pericyte markers that you'd look for. So in terms of the optimized protocol overall, what we've done is run through a variety of different combinations of cell concentration, endothelial cell to astrocyte to pericyte, and generally using this protocol. So we start out day 0, we seed the device, we set up an interstitial flow through the gel region in order to help establish the vascular, the BBB model. We then sometimes would apply a monolayer on the sides of the gel in order to just get a better seal so that we can get more accurate measurements of permeability of the BBB model. And then we would apply bidirectional flow, one way, first one way, and then the other day, alternating on days. And then starting on day 10, we'd apply unidirectional flow. And then by day 12 to 14, we would start to do our studies, which include permeability studies, immunofluorescence, sequencing, and so forth. So this is the cell ratio that we came up with, 1 to 0.6 to 0.08 for the endothelial cells, the astrocytes, and the pericytes. And you can see here you get a nice, well-structured three-dimensional vascular network that can be perfused completely. Here we've perfused in a fluorescent 150 kilodalton dextran, and you can see the fluorescence here that shows the filling of all the vessels. So we characterize this in a number of ways. Here we've looked at the morphology and looked at the consistency of the morphology. We've looked at the number of branch points, the number of segments, the mean segment length, and the diameter. And you can see that among these 5 different experiments, we saw very little variation from one experiment to the next. And in particular, one thing I want to point out is that the diameters here, they're a little larger than you'd expect, but they're also probably larger than the actual effective diameter because this is based on a 2D projection. and the vessels tend to have kind of an elliptical cross-section. So the vessels have good morphology. We also looked at the immunofluorescence of different factors, including ZO1, for example. ZO1 is one of the important junctional proteins in the BBB. Here you can see it's highly expressed in these cells, and it tends to localize at the junctions, which is what we would expect to see. Also VE cadherin, the CD31. And looking at the pericytes, again, using immunofluorescence, what we're looking at on the left-hand side is we're zooming through a 3D stack that shows the PDGFR beta is actually the purple here that shows up. The UEA is a membrane dye for the vasculature, and you can see it also here. You can see the cross sections of the vessels. The pericytes tend to wrap around these vessels and tend to be very much associated with the vessels. So we're recapitulating that aspect of the BBB in the sense that there's a close association between the pericytes and the endothelial cells. We can also look at the astrocytes, and we see good evidence of the astrocytes, again, interacting with the endothelial cells. Here, GFAP is used as the label, as the antibody to label the astrocytes. And we see evidence of astrocyte end feet that are connecting up with the vasculature and interacting with it. Working with a collaborator here at MIT, Seshin Yu, and her student Kunzan Liu, we use this technique of SLAM imaging in order to go deeper into the tissue and to use a label-free method that enabled us to look at the different cell types within this IPS-based model. Because they were not fluorescent cells, we wanted to be able to identify and do the cell count as a function of distance. So here's an example of one of the images on the left. And here you can see this goes in about 500 microns, which is the total height of the device. This is inverted. This is the cover slip down here, and then this is the top of the device down here. And by using these different metabolic measures, FAD, NADPH, and then also using the triple harmonic generation intensity of the signal, we found that we could cluster the cells. When we went in individually and identified the cell type from its morphology, we could associate it with a particular point here, and found that the pericytes, the endothelial cells would cluster in the middle here, the pericytes would cluster over here, and then the astrocytes would cluster here. So even without having fluorescent labels, or we've actually confirmed this though with markers for the different cell types, but we've been able now to convince ourselves that we can get the cell count as a function of depth once the system has fully developed. And here you can see distribution of the cell number. You can see that at the bottom of the device, we tend to get a higher cell density just because of the settling of the cells. But through most of the range of the device, we're getting the cell ratios that agree fairly closely with what you see in vivo. I see there's a question. Maybe I'll save the questions for the end if that's okay. But please feel free to put them in as they come to you. And I'll open up the Q&A box at that point. We also looked at barrier integrity within our BBB model. Here we've done is we've looked at a variety of different size dextran molecules and found that we could discriminate very clearly the permeability going from 150 kilodalton all the way down to a 3 kilodalton. These are fluorescent dextrans. And the measurements that we get, for example, for the 10 kilodalton dextran, you can compare to measurements made in rat brain capillaries. And you can see we're actually getting a slightly lower permeability. In other words, it's a slightly tighter barrier than measured in vivo. But you have to keep in mind that it's very difficult to make these measurements in vivo. And there's a fair amount of scatter in those measurements. These images just depict basically how we make the measurement of permeability. We introduce the dye, the fluorescent molecule inside the vasculature, And at a later time, we observe how much is leaked out. So then we can calculate how leaky those vessels are. We can also look at factors that are known to alter barrier function. Here we've looked at something called mannitol. Mannitol tends to loosen the cell-cell junctions between the endothelial cells. And as a result, the permeability increases. So we were able to demonstrate that our model exhibits that. Again, it increased, in this case, looking at a 70 kilodalton dextran, that's what these measurements correspond to, a significant increase in the permeability. We could also look at permeability, not just from the vessel inside, but also from the tissue, the brain tissue on the outside going into the vessel. And that was important to us because we wanted to be able to measure the permeability of amyloid beta going into the vasculature that's being generated by the neurons inside the brain. So here what we did is we did sort of the opposite. We developed a situation where we had the fluorescent molecule flooding the extracellular space, washed out the vasculature, and then we looked at the molecule leaking into the vessel. And then we can measure the permeability both ways, vessel to matrix, matrix to vessel. And what we found at that for 40 kilodalton dextrans and also a number of other molecules that we tested, that there were no differences between the two. Keep in mind that the other reason we want to do that is we want to test directional transport with the different transporter proteins, because some transport from the brain tissue into the vasculature and others transport from the vasculature, the intravascular space, to the extravascular space. So we wanted to further test some of the active transport modalities that are present in the brain. Keep in mind that the brain BBB is extremely tight, so it keeps most molecules out of the brain, but there are certain molecules that are needed, and for these, the brain typically has a number of different transporters that mediate this receptor-based transport. There are a number of different mechanisms that can be used. And these are also, by the way, used to get drugs or large cargoes into the brain, for example, with nanoparticle delivery. Transferrin, for example, is one, angioPEP2 is another. Different ways of getting optimal transport of drug into the brain. So currently, we find that there are a number of promising approaches, especially for nanoparticle drug delivery, with these receptor-mediated transcytosis shuttles. In particular, we looked at the transferrin receptor 1. It's one of the principal RMT targets, and it's expressed highly in the brain on the luminal side and is effective in transporting whatever you're interested in getting into the brain. So we looked to see, first of all, whether the transferrin was expressed in our model, and it certainly was. And you can see that it's expressed in the right location. Here the transferrin receptors shown in red. We could also actually see in high resolution imaging that it was on the luminal side of the vessel rather than the abluminal side, and that it tends to be expressed not homogeneously in the endothelium, but expressed in a large number of the endothelial cells. We also looked to see if we could demonstrate the transport via the transferrin receptor. So first of all, we measured the permeability of transferrin by itself. We then saturated the system, saturated all the receptors with non-fluorescent transferrin, and then did the same experiment. We saw a reduction. And by comparing to a 70 kilodalton dextran, which is the same molecular weight as about the same molecular weight as transferrin itself, we can show that the transferrin is being actively transported and it has a higher permeability. We also looked at an IgG molecule that's functionalized with the transferrin receptor. Here, this MEM189, it's a mouse IgG that's functionalized with transferrin. And you can see here we get an increase by about a factor of 10 in the permeability as a result. For comparison is just human IgG. going back to the SLAM imaging that I mentioned before, the other thing we can do with that is to actually measure in a time-resolved way the transport of molecule into the tissue from the vasculature. And the way we do this is to, first of all, and this is work that was done by a team of postdocs and grad students in collaboration with CISH&U. And I really want to emphasize the fact that these three postdocs, Sarah Spitz, Francesca Promaton, and Zhengyu Zhang, did a tremendous job in gathering all the data that essentially that I'm showing you today. In this particular case, we could look at the transport of transferrin from the vessels out into the surrounding tissue as a function of time. So this is a movie showing the accumulation. This is over a 1 hour period. And you can see that the distribution of transferrin is very patchy. That actually is showing the fact that transferrin is being taken up not just into the extracellular space, but also being taken up by the resident cells. And the other advantage of this technique is that we can identify individual cells. I showed you how we could identify the cell types before. So we can say to what extent the transferrin is being taken up by the endothelial cells. It's known that a large part of it tends to be, especially for short times, and less taken up by the pericytes and astrocytes and getting into the matrix. But we can now start to discriminate between, where the drug is going. In this particular case for transferrin. So if we look at, this is just images, a split image, this is the time equals 0, and this is time of 50 minutes. We can also look at individual cells. So here we looked at two different endothelial cells, one that was wrapped by a pericyte, that's the one in blue, and another one that was not surrounded by a pericyte immediately. And here you can see the two cells. But we found an interesting observation that the transferrin uptake tended to be higher in cells that were not wrapped by pericytes, suggesting that there's kind of a selectivity of uptake by the endothelial cells. Here we can see uptake of two different pericytes. We also find that near the end of this period, it looks like there's an increase in the amount of uptake that's occurring in this PC1. We could also look in astrocytes, and this was a particularly interesting finding where we saw in one part of the astrocyte, this region 1, we saw that there was an accumulation of the transferrin. So we can not only look at what cell types are taking up the drug, but we can also see where within the cell that drug is being deposited. We've also developed methods recently that enable us to measure the permeability of unlabeled compounds. And I'll quickly go through this. We basically use the same device that I was showing you before, but then once we introduce the drug, let it incubate for 9 minutes, they then extract the gel, enzymatically digest it using liberase. Then we can separate the extracellular matrix component from the cellular component, spin down the cells and separate them, and measure separately how much is taken up by the matrix and how much is taken up by the cells. Using this technique, first of all, we can measure what the permeability is. These are the measurements of permeability for this MEM189 that I mentioned to you before. These are from confocal measurements with a fluorescent version of the molecule, and these are ELISA measurements. And you can see we're getting good agreement between the ELISA measurements and confocal. This is the equation that we use for measuring the permeability from ELISA. And here just shows one of the variables that we have in here is the surface area. And there's some degree of variability, but the coefficient of variance is only about 20%. An advantage of this technique with the unlabeled molecules is that we can separate the matrix from the cells and measure the amount of uptake of the drug in the two compartments. So here we have several different experiments. The purple corresponds to the unlabeled transferrin, and then we've got labeled transferrin. The blue box is here. This is the MEM189, just two different fluorescent tags on them. The blue, turquoise, and the orange. And in these measurements, what we found is that we could then separate and identify the mean concentration in the cells and the mean concentration in the matrix and see how much a ratio between the two, which in this case turns out to be, for when we start talking about drug uptake, this is going to be considerable interest. So I'm going to move on to talk a little bit about the amyloid beta transporters in our system. Here we're interested primarily in RAGE, LRP1, PGP, and BCRP. And here's where they're located, either on the brain side of the endothelial cell or on the blood side. This just shows that each of these is expressed in the endothelium of our model. Again, the BCRP, LRP1, PGP, and RAGE. We can also show functionality of the transporters. Here you can see we've used alacridar with docetaxel as a substrate and PGP inhibitor, alacridar, and showing that we can distinguish with and without the inhibitor in terms of transport rates. And so I'm going to switch gears now and in the last, maybe the last 5 minutes, I guess, talk about the work that we've been doing combining the blood-brain barrier that you've just seen and that we've characterized in a number of different ways, adding to that now the Alzheimer's or Parkinson's organoids. For these organoids, we got the iPS cell lines from Jack's labs. They have modified them so that there's one with a single point mutation for Alzheimer's or a separate one that has a single point mutation for Parkinson's. I'm going to be showing you primarily the results from Alzheimer's disease. We take these iPS cell lines and differentiate them into cerebral organoids for the AD model or midbrain organoids for the Parkinson's model. And we also, for other experiments, differentiate them into neurons. And these are the protocols that we use for these. For the organoids themselves, the organoid protocol takes about 90 days. That's done in the Whitehead lab. They then provide us with the organoids. We can insert them into the central region of the device, and then we can observe them over a period of time. So this is the protocol that we use for the organoid differentiation. It's about a 3-month protocol to get the cerebral organoids. We're also going to add microglia to the system. And here's, again, the microglia are being obtained from the iPS11 cell line. A relatively short protocol, 6 to 10 days. And we combine the microglia then with the organoids and then insert those microglia containing organoids into the blood-brain barrier model. These are just some studies showing that the neurons show high levels of A beta 42 and 40, and that those levels are altered by a beta secretase inhibitor, or BSI. Here's with and without the BSI. So they're behaving in the manner that we'd expect them to. These are some 2D images. This is the single nucleotide variant. This is the Alzheimer's model. And then this is the single point mutation is corrected. And that's what's referred to as the control or the reverse here. This is what the organoids look like. That we're using MAP2 for the neurons, GFAP to indicate the astrocytes, and then DAPI for all the cells. And here you can see the top, this is the non-Alzheimer's model. This is the Alzheimer's model below. The things that you note immediately are that, one, there tends to be higher level of astrocyte deposition or growth in these organoids, and also fewer neurons, and also the activation of the astrocytes tend to be higher. We've also, in adding the microglia, basically the procedure that is used is to take the organoids, slice them in half, seed these differentiated microglia on top of those organoids. They then close back up, and then they can be inserted into the microfluidic system. And here's an image of the microglia under the control circumstance, and then also in the AD model. And here you can see the microglia migrating away from, out of the organoid, which would be over here in this case, migrating out into the matrix surrounding the BBB and interacting with them, and probably giving rise to some of the change in permeability that we see here between the normal and the Alzheimer's model. So we can see some phenotypic changes now, even in this simple model that we have with the single nucleotide variation. So just to summarize with talking about future directions, we're looking at making patient-specific models. We are working on adding the different immune cells to the system, both the adaptive and the innate immune systems. We've already added the microglia. We can certainly add the macrophages, and are looking to include other circulating immune cells. Here's a case, for example, in a previous model where we've introduced neutrophils into the system around a couple of cancer cells. And we can add continuous physiological flow, which we've done in a number of other experiments. We would like to include new technologies, and in particular, looking at ways of monitoring the health of the neurons, the neural activity in the device. MEAs, various approaches to MEA, but in 3D systems, are things that we're looking at right now. We're also interested in developing computational models, or essentially a digital twin of our system, so that we can capture a wider range of behaviors. And then also we're working on new higher throughput versions of this IPS-derived microphysiological BBB and cardiac organoid co-culture. And for that, we're working with my company, AIM Biotech, using this organics plate. So with that, I'd like to finish up just by acknowledging the people who did all the work that I've really just described. Our wonderful collaborators, the Sishin Lab, the Yanish Lab, Maya, who runs the stem cell facility at Whitehead, some of our funding sources, and in particular, a lot of this work was funded by industry, particularly Takeda and Daiichi Sankyo. From the Sishin U Lab, Kunzin Liu and Hanghao Cao are students that worked with us closely in using the SLAM techniques to make the measurements that I showed. And then finally, a real shout out to Francesca, to Sarah, and Zheng Yu, who really carried the bulk of this work. And with that, I will stop sharing my screen, and I would be happy to answer questions.
Roger, thank you so much. This was really, really inspirational. It was a fantastic lecture. So you got a lot of questions, and I hope that you will agree to answer offline some of these questions that we cannot address due to we have only 6 minutes. We will give you contact information. So next week, just before we switch to questions, we will have Kent Leach, who will speak about something completely different, which is engineered biomaterials to regulate inflammation and enhance tissue formation. And Kent works in a musculoskeletal complex. So Roger, maybe you can start from Jacob Schimmelmann's question. He says very clearly how much you influence the entire area of their work, and he's interested. Do you have an explanation for the poor vessel infiltration into the brain organoid tissue? So this is your biomaterials paper that was published.
Right. Yeah, and it's a great question, and it's something that we're working very hard on right now. Turns out that the vessels don't penetrate well into matrigel, which is what the neurons were seated in the previous paper, and which we're still using for the organoids now. We're exploring a number of different defined matrices that we can use that are both receptive to formation of the BBB, but also are not toxic and tend to facilitate culture of the neurons. We have some candidates right now that we're working on, but it's a little bit too early to say whether or not those are going to work out. But it's something that's certainly been on our mind for some time. Thanks for the question.
Okay, so two of attendees, Rulin Zhang and Amirali Selahi, are asking a couple technical questions, and they are, why is bidirectional flow important?
Mhm.
What is the throughput of the devices? Is the device automated, and to which extent? Um, and are there any plans to further increase the throughput and add automation, etc.? So maybe you can just elaborate a little bit about like where you are at this point with.
Sure. So in terms of the bidirectional flow, what we found was that, and Sarah and Francesca found this, I say it's a royal we here. Um, they found that if they just used unidirectional flow from the start, that you tended to get asymmetries in the development of the BPP. And they found that by reversing the flow direction during these first few days of culture, that they could eliminate that. So very just essentially an experimental finding. In terms of higher throughput, um, so I alluded to that a little bit at the very end of my talk. We are looking to see if we can develop the same BBB model in the organics chip that AIM Biotech makes. We're just about there. We've been working on that for a few weeks now, and we have been quite successful, I'd say, at developing the BBB. Shouldn't be difficult for us then to add the brain organoids to that same system because it also has a port at the top that we can introduce. That would increase the throughput to 24 experiments on a 384 well platform. Um, we, I guess the one challenge there is that we won't be able to generate very large interstitial flows, and we have to see whether the BBB will develop properly without them, with lower levels. Um, let's see, was there?
I think this was it. Yeah. Uh, it was something
Oh,
about automation, also like especially
yeah.
with
So again,
imaging.
with automation, if we can get this system to work in the AIM chip, that is compatible with the various robotic systems. In fact, OpenTrons, for example, is one that I know that they've worked with. So it should be able to automate the experiments.
Okay, so Wen Chao Gu had a question about ETV2 derived endothelial cells that you studied in vitro. Uh, is there an issue of uncontrolled angiogenic sprouting and network instability when you are in vitro?
Mm, that's an interesting question. So what we found we had to do when we first differentiate these cells, they're very highly proliferative, which causes problems. So we actually have to passage them out 4 or 5 times before we use them in our system. By that time, the proliferation rates are lower, and we don't then get the uncontrolled angiogenesis or sprouting that we might get otherwise. But that's a good point.
Okay. And then we are like almost at the top of the hour. You have a lot of questions. Hope you can answer them later, like another 11 or so. And let's finish with the question asked by Pam Graney. So she's interested if you could comment on the role of the lymphatics in the neurodegenerative diseases that you are studying, and if you have considered some ways, methodologies to incorporate the lymphatic vessels into your system.
Yes, we have. And in fact, we should be coming out with a publication on that fairly soon. What we've done is we've taken the system that you've seen, but now on one portion of it, we sprout lymphatic cells, a lymphatic network into the gel. And we use that then as a means of looking at interactions and clearance of A beta from the system. That system actually works quite well. We've published on the lymphatic vasculature a couple of papers over the last few years, but we now have a system and it's up and running, and we've been using it now for, at least a year now, that has the lymphatic system in combination with the BBB and the neurons.
Thank you, Roger. We will definitely look out for this paper. So thank you again for really, really most interesting question. And I would like to thank the attendees for very insightful questions that you are asking. And everyone will get the response directly after the lecture. Thanks,
Great.
everyone, and see you next week.
Bye-bye.